And if I do that, we've already succeeded.
Yes.
This is great.
Perfect. Success. Yay.
So, so one qualifier, I did let Holly know that what we work on is not intimately related with reproduction, but hopefully it's got a lot of immunology in it, and hopefully you will find this talk of interest. So, so the title of the talk is Multiple Sclerosis, Animal Modeling, and the Role of the Innate Immune System in Autoimmune Neuroinflammation. So what I'd like to do is first start off by acknowledging the people that have contributed to the work in the past and are currently doing the studies. So Dr. Hongwei Chen, there with the red circle around her face, is an associate professor in the lab, and she started the project that I'll be speaking on. And two former graduate students, Yudong Liu and Chaoqi Yan, and Chaoqi is also shown in that one picture with the red circle, have driven the work. And then shown here, let me make sure. So we had 3 new lab members join in 2020 during the pandemic. We usually take a group photo every year, and we couldn't do that last year. 3 new people joined, and one, Bill Turbot, shown here as a postdoctoral fellow in the lab that is continuing these studies. So what I'll do first is give you some background to multiple sclerosis and its animal model experimental autoimmune encephalomyelitis, or EAE. Then introduce you to the signaling pathway that we've studied for many years, the JAK-STAT-SOX pathway, and then show you the data on the role of innate immune cells in brain-targeted EAE, some conclusions, and then a little bit of discussion about next steps. So multiple sclerosis is an autoimmune disease in which immune cells target the myelin sheaths that surround nerve fibers, leading to neurological disturbances. Like many autoimmune diseases, it is much more prevalent in females to males. And again, like many autoimmune diseases, prevalence has increased significantly over the past decade. And currently, there are 1 million people living with MS in the United States. So as mentioned, it's an inflammatory demyelinating disease of the central nervous system, resulting in motor, sensory, and cognitive dysfunction. On the positive side, there's actually about 20 FDA-approved disease-modifying therapies for MS. These are, for the most part, immunomodulatory. However, none stop disease progression. The pathological features of MS include the infiltration of immune cells from the periphery, demyelination, which is caused by loss or cell death of oligodendrocytes, gliosis, which is a hyperproliferation of astrocytes, lesion formation, and neurodegeneration. The symptoms of MS are shown on this slide, and you can appreciate that many in addition to the brain, other organs are affected. So what I'm showing on this slide is the latest classification of multiple sclerosis. Relapsing remitting MS, shown on the bottom, is the most common form of MS, unpredictable attacks, which may or may not leave permanent deficits, followed by periods of remission. This can then transit into secondary progressive MS, where the relapsing remitting form begins to decline without periods of remission and relapses. And perhaps the most devastating form of MS is primary progressive MS, with a gradual progression of disease from its onset with no relapses or remissions. And what I should add is that all of the FDA-approved drugs are effective for patients with relapsing remitting MS, but only one of them actually is effective for patients with primary progressive MS. So to this day, we still don't know the etiological cause of MS, but what does happen is activation of immune cells in the periphery, leading to a breakdown of the blood-brain barrier and the infiltration of these cells into the central nervous system. So these include innate immune cells such as macrophages and neutrophils, T cells, both CD4 and CD8 positive T cells, B cells, plasma cells, and dendritic cells. In addition, there's an imbalance of what we call pathogenic T cells. These are T cells producing cytokines such as gamma interferon and Th17 and IL-17 to a population of cells known as T regulatory cells, which are anti-inflammatory in nature. Now, as I mentioned, there's a number of alterations of the endogenous glial cells, hyperproliferation of astrocytes, damage or loss of oligodendrocytes leading to demyelination, and activation of the endogenous macrophage of the brain, the microglia, leading to immune activation. It's also appreciated that there is significant neuronal damage, axonal loss leading to neuronal death, which contributes to some of the neurodegenerative aspects of multiple sclerosis. In addition, there is aberrant cytokine and chemokine expression, particularly those with pro-inflammatory properties. And what I'd like to emphasize is that there is tremendous heterogeneity of both the immune cells and glial cells in the diseased central nervous system. As a nod to the audience, I think it's well known to you in the audience that there is a correlation with pregnancy and MS. Well documented that there are fewer MS relapses during pregnancy, especially in the third trimester, where there's actually about a 70% reduction. So changes that take place during pregnancy are thought to contribute to reduced inflammation, less MS activity, and fewer relapses. And these include elevated levels of estriol and reduced levels of pro-inflammatory cytokines such as IL-12, TNF, and interferon gamma, as well as an increase in anti-inflammatory T regulatory cells. So now I'll introduce the signaling pathway that we're interested in. This is the JAK-STAT pathway. Just gonna try to use this here. The JAK-STAT pathway that's used by over 70 different cytokines, interleukins, interferons, and growth factors for signal transduction. And the activation of this pathway is really critical for the initiation and regulation of innate and adaptive immune responses and ultimately termination. So just to walk you through this, a cytokine of interest will bind to its receptor, and associated with the receptors are Janus kinases. These are tyrosine kinases. These JAKs will phosphorylate tyrosine residues in the receptor intracytoplasmic domain. And recruited then are transcription factors known as STATs for signal transducers and activators of transcription. They are recruited to this JAK receptor complex. They in turn become tyrosine phosphorylated, translocate into the nucleus, and turn on genes that are essential for both innate and adaptive immune responses. What we do appreciate, however, is when this JAK-STAT pathway is not regulated, and by that I mean it's not terminated. This has pathological implications, particularly for autoimmune and inflammatory diseases and many cancers. So to keep the JAK-STAT pathway in balance, a number of mechanisms have evolved to negatively regulate this pathway, thereby attenuating biological responses. And we'll focus on one family of these negative regulators, and these are known as suppressors of cytokine signaling, or SOX proteins, that function as negative regulators. So I described to you this signaling cascade where phosphorylated STATs bind to receptor domain, or excuse me, there we go, back. Okay, so they bind to regulatory elements and genes important for the immune response, but they also actually turn on their own negative regulator, the SOX family of proteins. And these SOX will inhibit the ability of JAK kinases to tyrosine phosphorylate, essentially terminating this response. And just to add here, there is a family of small molecule inhibitors known as JAK-inhibs, similar to the SOX proteins. They target the JAK kinases to therapeutically inhibit cytokine signaling. There are about 4 JAK-inhibs that are FDA approved right now, and they're used in a variety of cancers, autoimmune and inflammatory diseases, and most recently in the context of COVID-19 infection to suppress the cytokine storm. So with respect to these SOX proteins, sorry, my cursor is not functioning well. As I mentioned, these are cytokine-inducible proteins. There are 8 family members. They inhibit signaling through the JAK-STAT pathway by a number of mechanisms. And we will focus on one family member known as SOX3. Okay, so what I want to do here is just present you with some evidence of dysregulation of the STAT-SOX axis in MS and its animal model EAE. So first of all, it's been shown that both STAT3 and STAT4 are MS susceptibility genes that are associated with an increase in MS risk. And monocytes from patients, MS patients with relapsing remitting disease, have heightened STAT3 activation and less SOX expression during relapses. It's also been shown if one looks in active MS lesions in the infiltrating myeloid cells that STAT3 is activated. And it's also been shown that CD4 positive memory T cells from MS patients have higher levels of activated STAT3 and STAT4, which drives the induction of pathogenic T cells. And it's also been shown in the context of the generation of pathogenic Th17 cells. These are the cells making the cytokine IL-17, that STAT3 is absolutely required for the induction of pathogenic T cells. So we've previously shown, using some of those JAK-inhibs that I spoke about earlier, that inhibition of the JAK-STAT pathway ameliorates disease severity in a number of EAE models, in part by inhibiting STAT3 activation. And then regarding the SOX proteins, we and others have shown that SOX 1, 2, and 3 are critical for regulating inflammatory responses in astrocytes, macrophages, and microglia. So based on a number of years of work in the lab, we had a question, and that was, does hyperactivated STAT signaling in myeloid cells contribute to autoimmune neuroinflammation? So how can you model that? First of all, we knew that the genetic deletion of SOX3 was embryonic lethal. So what we did was to generate mice that were deficient in SOX3, particularly in cells of the myeloid lineage. So we crossed SOX3 flox mice with mice, Lysem-Cree mice, and this will generate a complete deletion of your gene of interest in both bone marrow-derived macrophages and neutrophils. So we generated these mice and did a lot of characterization of the cells and of the mice, and I should say we initially focused on the macrophages. What we noted is that there was elevated STAT3 activation, and of course this makes sense because the negative regulator of STAT3, SOX3, is lacking in the cells. And we did an extensive characterization of SOX3 deficient macrophages and found that they were polarized to what we would call a pro-inflammatory phenotype, expressing high levels of class 2 MHC, many pro-inflammatory cytokines and chemokines. These cells could induce the differentiation of pathogenic Th1 and Th17 cells, and also could themselves induce death of neurons. So with these mice, we subjected them to EAE. So this can be done in a variety of ways, but I'll show you data with active EAE. So this is where you immunize mice with particular neuroantigens. In this case, we use MOG, which is myelin oligodendrocyte glycoprotein. So the mice are immunized, and then they get pertussis toxin to help open the blood-brain barrier. And they exhibit clinical signs as shown in the right-hand panel. And this is classical EAE. So this manifests as ascending paralysis with preferential immune cell infiltration in the spinal cord. So that is classical EAE. So now we take our mice lacking SOX3 in myeloid cells, as well as the control mice, SOX3 floxed, and we subject them to active EAE. So when we look for signs of classical EAE that I just described, you can see that the control mice have disease as would be expected. What was of surprise to us is that the mice lacking SOX3 had absolutely no signs of classical EAE. Rather, they exhibited a different type of EAE that's called brain-targeted or atypical. The clinical symptoms are quite different. They involve ataxia and tremors. And rather than spinal cord involvement, the involvement of the cerebellum is very prominent. So this was actually quite a surprise to us, interesting, and so we have followed it up. So this is just the EAE scoring system, and I think you can appreciate just the sort of differences in clinical manifestations of classical, where spinal cord is involved, and brain targeted, where the cerebellum is involved. So work throughout the years, we've characterized these mice. There's extensive inflammation in the cerebellum, pronounced demyelination and axonal damage. We've looked at the activation status of the STAT transcription factors in the cerebellum and have shown heightened activation. The infiltrating macrophages and resident microglia are polarized to the pro-inflammatory phenotype. There is infiltration in the cerebellum of pathogenic Th1 and Th17 cells, B cells, dendritic cells, and most pronounced was neutrophil infiltration. So neutrophils are the first cells to infiltrate the cerebellum and the most numerous. And based on these data, as well as some other data, we really suspected that neutrophils were important in the brain-targeted EAE phenotype. And Yudong Liu, a former graduate student, performed this experiment where he depleted neutrophils with an antibody cocktail. And again, I think what you can appreciate here, so here is the atypical or brain-targeted EAE, which is delayed in onset and has a significant reduction in clinical symptoms when neutrophils are depleted. So if you haven't thought about neutrophils in a while, let me just tell you about them. They're actually quite fascinating cells. Although interestingly, they historically were really thought to just function as short-term effector cells with no long-lasting impact on immunological responses. And in fact, there is a phrase that these neutrophils functioned as rapid, mindless aggressors of poor regulatory function. So actually, that's not true. They're really important cells. Functions include phagocytosis, degranulation, production of reactive oxygen species, neutrophil extracellular traps. They produce many cytokines and chemokines and can actually function as antigen-presenting cells. They can also modulate other cells of the immune system in terms of either activation or suppression, depending on disease context. And what we now know really from studies in tumor settings is that there is phenotypic diversity of neutrophils due to transcriptional plasticity. And single-cell RNA-seq experiments have in fact revealed extensive neutrophil heterogeneity, especially in tumor settings. So we asked the question, what is the mechanism by which neutrophils induce brain-targeted EAE in mice lacking SOX3? And so in panel A, what we first show is demyelination occurring in the cerebellum. And in panel B, these are neutrophils that have been isolated from the cerebellum. And I should mention, this is all at peak of disease, where we're staining for a number of surface antigens. And we see an increase in CD11B and CXCR4, which is a chemokine receptor, and decrease in CD62L and CXCR2, another chemokine receptor. And this phenotype is actually reflective of primed or aged neutrophils that I'll discuss on the next slide. Again, we document the increase in neutrophils in the cerebellum in panel C, enhanced degranulation by these cells in panel D, and active production of reactive oxygen species by neutrophils. So as I mentioned, this phenotype that we assess by staining for surface antigens is representative of a sort of a subset of neutrophils called primed or aged neutrophils. These are overly active neutrophils associated with increased pro-inflammatory activity and enhanced ROS production. And in fact, circulating neutrophils from MS patients are actually characterized by this phenotype. So then the question became, what is contributing to this primed neutrophil phenotype in neutrophils that lack SOX3? So what we did in panel A was to first look at just wild type neutrophils and stimulate them with a variety of cytokines that activate the JAK-STAT pathway. And in this case, we're actually looking at SOX3 as a readout gene. And what you can see is of all the cytokines that we tested, GCSF, which stands for granulocyte colony stimulating factor, was by far the most potent activator of SOX3 expression in neutrophils. So what we did then was to stimulate either our control neutrophils or neutrophils lacking SOX3 with IL-6 and GCSF. And here we're measuring the activation of STAT3, looking at tyrosine phosphorylation. And you can see that neutrophils lacking SOX3 have enhanced STAT3 activation, and not shown in this slide. Not only is it enhanced, it is prolonged in duration. So these data suggested that GCSF, in fact, may be an important contributor to the activation of neutrophils. And we did a lot of experiments to substantiate this, but this is perhaps the most important experiment where we actually use a neutralizing antibody to GCSF to deplete it. And again, I think what you can appreciate is in mice that got the antibody treatment to deplete GCSF, there is almost a complete diminution of clinical symptoms. And importantly, demyelination is prevented. In addition, we see that in the cerebellum of mice with almost no disease, neutrophil infiltration is inhibited, as is the infiltration of monocytic cells. So another important feature of neutrophils is their ability to produce what are called extracellular traps or nets. Most people are familiar with nets in the context of having a sort of antimicrobial function. These nets contain antimicrobial peptides and are very efficient for the killing and clearance of extracellular bacteria, as well as parasites and other pathogens. So this is actually a beneficial function of net formation. However, we now appreciate that nets have been found in a number of autoimmune diseases, including lupus and rheumatoid arthritis, and in this context actually promote inflammation and organ damage. So in fact, a detrimental feature of net production. We know that neutrophils from MS patients display enhanced production of nets, but the functionality is not known. And whether nets play a role in the pathology of either MS or EAE is not clear. Now, what we looked at here, and this is work done by Chao-Chi Yan, is he asked whether there is evidence for net formation or netosis in the cerebellum of the mice that have the hyperactivated neutrophils. So the first thing, if you could appreciate, we're staining with Lys6G, which identifies neutrophils, and you can see very few in our control mice with lots of neutrophils in the cerebellum of mice lacking SOX3. And when you can look for nets by staining for citrinated histone 3. And I think you can appreciate that a subset of the neutrophils are in fact producing nets, which is not seen in our control cerebellum. So we do have evidence for netosis. We still don't know the functional implications, and that's something that we're currently working on. So to summarize that data to date, looking at SOX3 deficient neutrophils that come from the cerebellum, they produce high levels, again, of many pro-inflammatory cytokines, chemokines, and mediators such as ROS. They also produce complement components C1q and C4. They have a heightened responsiveness to GCSF, and they display prolonged activation of the STAT3 pathway. And phenotypically, they express markers that are reflective of primed or aged neutrophils. In vivo, we've shown that depletion of neutrophils suppresses disease onset and severity of brain-targeted EAE. In data that I didn't show you, ROS scavenger treatment also reduces disease severity and onset, and showed you that treatment with anti-GCSF antibody could ameliorate brain-targeted EAE by suppressing neutrophil and macrophage infiltration. And what I should mention is the characteristics that we've defined of these SOX3 deficient neutrophils really reflect the phenotype of neutrophils from MS patients, including elevated degranulation, resistance to apoptosis, enhanced production of reactive oxygen species, and neutrophil extracellular traps. So these studies do implicate neutrophils as having a very important role in brain-targeted EAE. But there's one important caveat with all the data that I've shown you to date, and that is that we are looking at mice that have SOX3 deleted in both macrophages and neutrophils. So is this brain-targeted EAE the result of the neutrophils, the macrophages, or a combination of both? And so to be able to address that question, we generated mice with SOX3 deletion exclusively in neutrophils using a mouse that's called a ketchup mouse. These mice, so this is Lys6G neutrophil specific loci that's been modified to drive expression of both CRE and red fluorescent protein TD tomato, and that's why they're called ketchup mice. These neutrophils have been shown to be functionally normal and present in physiological numbers. And so we crossed our SOX3 flox mice with these ketchup mice and generated mice that have complete SOX3 deletion specifically in the bone marrow-derived neutrophils. And what I should mention at this point, as the breeding of these mice was going on last year, this is when COVID hit. And we basically had to pretty much, we kept the mice at the minimum level that was allowable at the time, but as you might imagine, experimentation rather ground to a halt. And then once we were able to get back in the labs, Bill Turbot was able to start the characterization of these mice. And obviously the first thing that he looked at is what kind of EAE do they have? So let's look in that middle panel, brain-targeted EAE, reminding you again, these mice have motor deficiencies and ataxia. And so here Bill is comparing mice, the Lys6G mice with SOX3 deletion exclusively in the neutrophils compared to our Lysem mice, which have SOX3 deletion in both neutrophils and macrophages. And you can appreciate that the brain-targeted EAE phenotype is virtually identical in both of those mice. And survival analysis also shows a very comparable profile. So as you might imagine, this was really gratifying, a very gratifying result, suggesting in fact that deletion of SOX3 in neutrophils was specific to generate this brain-targeted EAE phenotype. So Bill has gone on to start characterizing what is happening in the cerebellum of these mice, and I'll just show you one or two pieces of data. First, again, documenting that there are increased CD4 positive T cells in the cerebellum of the mice. And then he characterized, well, what kind of T cells are these? And we talked about pathogenic T cells. These can be Th1 cells producing gamma interferon, or Th17 cells producing IL-17, or cells that produce both IL-17 and gamma interferon, or cells that don't produce either, and those are naive. And what I'd like to just emphasize here in panel B, looking at the CD4 positive T cells from the SOX3 deficient mice, the majority of the T cells were actually of the Th1 phenotype producing gamma interferon, with a very, very small population of Th17 cells. This is actually different than what we see in the mice lacking SOX3 in both macrophages and neutrophils, where there's actually a predominance of Th17 cells and less Th1. So this is sort of an interesting difference that we're starting to observe. CD4 T cells have a lot of plasticity and can express a variety of cytokines and chemokines in conjunction. And so here, Bill's looking at the ability of these Th1 gamma interferon producing CD4 positive T cells to produce a cytokine known as GM-CSF. This is granulocyte macrophage colony stimulating factor. This is also considered to be a cytokine that contributes to the pathogenesis of EAE. And here we see that the Th1 cells from mice lacking SOX3 are producing higher levels of GM-CSF compared to their controls. So to summarize where we are with these mice, we've shown that SOX3 deletion that is specific to neutrophils is sufficient to induce the brain-targeted EAE phenotype. In data that I didn't show, we've also seen in these mice the increased infiltration of neutrophils into the cerebellum, and they have a similar primed neutrophil phenotype as described for the Lysem-Cree mice. CD4 positive T cells from the cerebellum from these mice skew towards a Th1 phenotype with increased expression of GM-CSF. So at this point, we're actually confident in saying that we believe that hyperactivated neutrophils, in this case as created by the absence of SOX3, can actually function as instigators of the brain-targeted EAE phenotype. And of course, further characterization of these mice is ongoing. So at this point now, let's go back to our SOX3 Lysem mice. And again, remind you, these are mice that lack SOX3 in both macrophages and neutrophils. And what we did was an experiment where we wanted to ask, what is the profile of the myeloid cells in mice with both classical and brain-targeted EAE? So what we did is we took our control SOX3 flox mice and induced active EAE and isolated myeloid cells from the spinal cord of those mice at the peak of classical EAE. And similarly, we did the same experiment taking myeloid cells from the cerebellum of SOX3 Lysem mice at the peak of brain-targeted EAE. And these were sorted CD4 positive, CD11B positive myeloid cells, and single cell RNA-seq analysis was performed. So what we found using unsupervised clustering was actually 5 different clusters of neutrophils, 2 macrophage clusters, 1 cluster of microglial cells, and 1 cluster of dendritic cells. So in panel A and B, showing you the different clusters, showing you the 5 neutrophil clusters, and then we have put together what we call the other myeloid lineage cells in gray. And if you look at C, what you can appreciate is now if we're looking at the spinal cord from the mice that have classical EAE, the majority of cells in the spinal cord are actually myeloid lineage cells. So these include infiltrating macrophages, dendritic cells, and microglia. They do have those neutrophil clusters, but they are not as abundant compared to mice where we're looking at the cerebellum of mice lacking SOX3. You can appreciate they have less infiltration of myeloid cells, the macrophages and dendritic cells, and a higher representation of infiltrating neutrophils with very prominent percentage of this K-nu1 cluster followed by K-nu2. So what are these different clusters? And again, just remember, this is single cell RNA-seq data. So what we're looking at here are transcripts. So this is reflecting mRNA expression. And you can see the genes that are represented by the different clusters. And there are certainly differences between the 5 neutrophil clusters. And so let's take sort of a closer examination at some of those that might be relevant to neutrophil functionality. In the NU1 cluster, we see expression of ICAM1, which is involved in neutrophil adhesion and migration. Another gene, PLAUR, involved in chemotaxis of neutrophils. In the NU2 cluster, we see expression of 2 genes, MT1 and 2. These scavenge free radicals. And Syndecan4, which is very important for the migration of neutrophils. In the NU3 cluster, we see CD9, which is a degranulation-related gene, expression of CD274, which encodes for PDL1, which controls CD4 T cell functions, and TLR2 expressed on this, the protein is expressed on the surface of neutrophils, involved in neutrophil adhesion and ROS production. In the NU4 cluster, we saw expression of CD177. This encodes for a receptor that controls neutrophil migration, and MMP8, matrix metalloproteinase, involved in matrix degradation. And in what we call the pre-NU cluster, we identified genes that represent type 1 interferon response genes, and gene PLSCR1, which is involved in GCSF-induced neutrophil production. So what we're currently doing is looking at the neutrophils from these mice and looking for expression, for protein expression of these genes of interest, and in particular, looking at expression of surface-related molecules so that we can actually sort and enrich for these different neutrophil clusters and do functional studies. So those preliminary data that I'm showing you here are of interest, but remember, we're just looking at peak of disease. So what we really need to do is to follow these mice in a longitudinal fashion, starting with mice, naive mice that have no disease, mice at the preclinical stage of disease, and then mice at the peak of disease. So the hypothesis is that intrinsic factors and environmental cues in the central nervous system will dictate the transcriptional profiles of neutrophil clusters, which in turn will dictate the functional properties of these cells. So we will, at this point, probably restrict these studies to the neutrophil-specific knockout and the appropriate control, look at naive preclinical and peak, looking at cerebellum and spinal cord, and doing the single-cell RNA-seq analysis, and hopefully being able to really look to see the trajectory of the development of different neutrophil clusters and what their functions are. And of course, so you've probably read a lot of single-cell RNA-seq papers, and they're interesting, but in my opinion, actually many of them are incredibly descriptive because they don't really tell you whether the genes that have been identified actually have any relevant function. And so you really need to look at protein expression of the genes of interest. And in our case, what we need to do is to evaluate the effector function of neutrophils that are relevant to neuroinflammation. So this would include functions such as net formation, the chemotaxis of neutrophils, their ability to produce cytokines and chemokines, and their ability to modulate other immune cells, in this case, CD4 positive T cells in terms of activation and polarization using enriched neutrophil clusters. And those are the experiments that are ongoing now. So in the context of EAE, neutrophils do have actually a very important role. We think they're involved in priming and activation of the immune system in the periphery, as shown in A. We know that they are able to first sort of disrupt the blood-brain barrier and then extravasate through the disrupted blood-brain barrier to take up residence within the central nervous system. So we know that they are key for disruption of the blood-brain barrier and for now promoting neuroinflammation within the central nervous system. We believe they're involved in myelin degradation, in phagocytic activities, and in modulating the functionality of pathogenic effector T cells. So this last slide, we've talked a lot about MS and the animal model EAE, but I wanted you to be aware that in fact neutrophils are implicated in a number of neuroinflammatory and neurodegenerative diseases, not only MS, but neuromyelitis optica spectrum disorder, Alzheimer's disease, Parkinson's disease, ALS, and ischemic brain injury. So in preclinical models of many of those diseases, neutrophils are considered to be pathogenic by the induction of neuronal damage and detrimental CNS immune and inflammatory responses. However, in some disease settings, protective and beneficial neutrophils have recently been identified that can inhibit neuronal damage and promote axonal regeneration. And we think this, sort of the bad and the good neutrophil is really reflective of neutrophil heterogeneity. And so our ultimate goal is to elucidate neutrophil plasticity, heterogeneity, and functionality as related to neuroinflammation. So Holly, that is my last slide, and I am happy to take any questions.
Tika, Holly had to sign up.
Thank you so much for a fantastic talk. Holly had to step out, but she left some questions.
Okay.
And I think, in the urogyne space for irritative bladder symptoms and MS, they typically would prescribe anticholinergic medications. So her question, she was wondering whether these immunomodulators used in treatment, and to your knowledge, whether they work as well, both centrally and peripherally. So
the immune modulators that have, that are approved right now actually do both. And I think what's asked about MS is it's sort of that chicken and egg question. There is certainly the inappropriate activation of immune cells within the periphery. And that's really what is thought to drive disease pathogenesis, clearly dependent on their ability to infiltrate into the central nervous system. So some of the drugs actually prevent the infiltration of immune cells into the central nervous system, and those are very beneficial. And then others just sort of down modulate the inflammatory response in myeloid cells, in T cells, and in B cells, and those are also beneficial. So I think probably it's most important to hit the target in the periphery, but of course, like all diseases, MS patients present with disease, and then you've got to treat it. So you've already got that sort of vicious circuitry ongoing.
Yeah, thank you. And I know there is a lot of interest around MS and reproductive issues at NICHD. They keep coming up with this, with these grants on just pregnancy and contraception, etc. Your last slide was great. It took off one of the questions I had on just what are analogous disease conditions where this mechanism may be at play. But I was wondering as well, upfront you showed the dramatic increase in MS over time, that incidence. And so how would you speculate on just what is driving this, and does this work provide any clues to that?
So what is driving the increased incidence in MS? Boy, I think if I knew the answer to that, I think it's very analogous to what we're seeing with so many of the other autoimmune diseases, rheumatoid arthritis, lupus in particular, IBD. And a lot of that is linked to the hygiene hypothesis, right? That we've gotten too clean over the years, and so we don't have enough sort of low level of infection, if you will, that sort of promotes a low level inflammatory or immune response. And then because of that, and the analogous, what people talk about is kids don't go out and play in the dirt anymore, right? They just have their play dates and stay inside and are very hygienic. So the sort of the lack of exposure to pathogens, which obviously, God willing, would not kill you, but get your immune system somewhat primed is totally missing. So when that insult comes, and what I should say about MS, there are environmental agents that are thought to promote MS. There is a genetic component. Again, we still don't know what causes the disease. Some people speculate viral infection during the teenage years, but it just seems like the immune system of patients that are susceptible, just with that first insult, just really over ramps and over activates. Now, does our work contribute to that? I would have to humbly say no. I don't think it does at this point.
Thank you. Really a great talk. Let's open it up to others. Are there questions? Or please feel free to speak up or drop questions in the chat box. Let's see. So hoping there is at least one more thing. I'll just step back and just look in general with COVID-19 and just how it's impacted what we do. So how did COVID-19 impact your work and perhaps the work of other basic scientists on campus, and what are the lessons at this point? We're still in it, but what are the lessons that we can draw and for the future?
That question. I could talk for hours. So I think for those of you that know me, so I'm the Senior Vice Dean for Research in the School of Medicine. And so I had the most unfortunate job of shutting down our research enterprise in the School of Medicine. And basically mid-March, lab activity ceased, with the exception of labs that were just starting to work on COVID-19 and the maintenance of the most essential animal strains. But for the most part, research, wet lab research was just shut down. I think probably some of you experienced that. And it was really, really hard. I think especially for the graduate students and the postdocs that are on a time-limited trajectory. So lab activity for the most part ceased in mid-March. And then we started to open up a little bit by the beginning of June. But if you remember at that point, very stringent social distancing requirements were put in place, as well as masking. And so people were allowed to start coming back to the lab, but so in shifts and think back now. I mean, so we're essentially in May of 2021. Think back to what we did not know in June. So there was huge trepidation, and one person would test positive, and how did you handle that, especially in a scenario of a building that has the open lab concept, right? So I would just say even though people were starting to come back, we encouraged people to only be in the lab for as limited a time as possible to do their experiments and then leave. And so I think it's fair to say from June, July, August, September, October, wet lab research was proceeding, but at quite a limited low-level activity. And things started to pick up a little bit in November and December. And then, of course, wonderful news with the vaccines. And where we are now is, I would say, almost back to normal. Social distancing requirements have been eliminated in the labs. Masking is still required. And of course, asking people to use common sense. So, it's interesting. There have been winners and losers in COVID-19, people working that could transition and pivot to working on COVID if you were a virologist. Lots of opportunities, lots of funding, lots of supplements. If you don't work on COVID, which is probably the majority of our researchers, lab work stopped. And what we're really trying to do is to get the NIH to provide cost extensions on all grants, because I think it's pretty safe to say about a year's worth of productivity has been lost with COVID-19. And we're not out of the woods yet. I mean, what's happening in India is far away, but it's not that far away, right? And it's very frightening. Alan, you're on mute.
I was just gonna, agree with you. Thank you for that insight. Are there other questions? All right. Well, Tika, thank you so much. I think it's a great opportunity to say thank you for your leadership and everything you've done to help us continue to navigate some very challenging times.
Well, it's my pleasure and delighted to be able to speak to the group. Thank you so much.
Thank you.
Thank you, Tika.
Thank you.
Bye.